Okay, so the first part of the cell cycle material that we'll be covering is what we will be going over this week. So first we'll start with an overview of this whole cell cycle. The cell cycle starts from G1 after the cell has already divided. The cell will go through a period of growth, and then during S phase, the cell will duplicate all of its DNA. And then in G2, the cell will begin to prepare for mitosis. And then the M phase is the entirety of mitosis, where the cell division occurs. And so we'll go through each one of the phases one by one. So G1 is the beginning of the cell cycle. And in order to continue in order to pass into the next phases, eukaryotic cells need to receive a growth signal in order to continue on. Otherwise, they'll stay in G1. G0 is a temporary or permanent state of non-division, but you don't have to worry about that for this class too much. So yeah, just focus on G1 right now. During this time, it's also going to, as how it's been growing, it's also going to check for DNA damage to see if it has enough nutrients and energy to continue to go on. And if it receives a growth signal and all of the nutrients and DNA levels are at the correct levels, the preconditions will be met, and then the cell will be able to pass through what is called the restriction point right here in towards the end of G1. And this is the point of no return, where it will continue into S phase and G2 and M. It'll go all the way through as long as it meets the other criteria. Yeah. So after it's gone past G1, the cell will be in the S phase, and this is the part where all of the DNA replication is happening, because remember, the cell only has one copy of its DNA. And so it'll have, in this right here, it'll make a duplicate so that it can split off later on, potentially, into two cells. During this class, we won't talk about it too much, but there's an S phase checkpoint. And actually, we'll talk about it next week, but when the cells can temporarily halt DNA replication if there are errors in the replication. And we'll also talk about mechanisms of repair later on. But all you need to know for now is that DNA replication occurs here, and that they check along the way for DNA damage before moving through to the next phases. And then in G2, this is the preparation just before mitosis. And so the cell is going to make sure that all the DNA has been replicated in the previous S phase, and to make sure that there aren't any major pieces of DNA damage that haven't been repaired yet from S phase, and to make sure they continue to grow and prepare more proteins, nutrients, and organelles to be able to split off. Make sure that both daughter cells, after splitting, would have enough to sustain themselves. And at the end of G2, there's a major checkpoint that will prevent cells from entering M phase until they have grown to the correct size and thoroughly checked all of their DNA. And then for this M phase right here, this is where the actual splitting occurs. During this phase, there is during this phase of the cell cycle, there are a few steps between G2 and getting back to G1. There is prophase where it's starting to break down the nucleus, as well as starting to prepare other stuff for the replication. Metaphase is when the chromosomes are lined up in the middle of the cell, as you can see in the diagram. Anaphase, the sister chromatids get split into two. Each side of the cell will eventually split off into two cells. So a copy of each piece of DNA is basically split on each side. Then in telophase, they start to reform and split off some of their, they start to reform like some nucleus, and they start to congregate again inside of at the poles. And then in cytokinesis, that's where the cell will like officially split off with the cleavage flow in the middle. And there's a major checkpoint as well within M phase that we'll touch upon more next week. But for now, just know that it's called spindle assembly checkpoint. And it's also called metaphase checkpoint because it prevents the cell from entering anaphase from metaphase. So it's basically the point at which the cell allows the sister chromatids to be pulled apart, quote unquote, pulled apart, as you'll see in the next week's like mechanistic breakdown of it. But for now, I just need to know that it prevents the cell from going into anaphase. But once it passes the checkpoint, it's allowed to pull those sister chromatids apart.
So the next part we're going to go over is the cell cycle control system. So the cell cycle control system is regulated by cycle activating and deactivating protein complexes required for initiation or regulation of the DNA replication, chromosome segregation, and cytokinesis. And I think for the most part, up until this week, we've always talked about phosphorylating, activating a protein, but that isn't always necessarily the case. As next week, we'll go over more in depth of WEE1, but it is mentioned in this week's topic, but WEE1 is a protein that will add a phosphate group inhibiting its target. And another quick note that phosphatases are what remove the phosphate group. And then the main kinase in the cell cycle control is CDK, or cyclin-dependent kinase. So these are serine threonine kinases. So they add the third phosphate group onto the serine threonine R groups. They're regulated by the cyclin levels, phosphorylation by cyclin activating kinase, which is CAC, phosphorylation, WEE1, which deactivates it. There are CDK inhibitors, and then an example of that is P21, which we'll go over towards the end of this presentation, and also by dephosphorylation. And then to activate CDK, two events need to happen. It needs to be bound to cyclin, and it also needs to be phosphorylated by the CDK activating kinase. So just binding cyclin is necessary, but it's not sufficient for it to be active. And then anytime we reach a checkpoint, cell breaks are put on by inhibiting or deactivating CDK. And also, if it's stalled for too long in any one point, like S phase, G2, whatever part, it's just gonna be signaled for apoptosis. It doesn't wait for it to get everything it needs for long periods of time. So like I said, cyclins are required for CDK kinase activity. They provide specificity for each phase in the cell cycle. So I think in the next slide, there's like cyclin E, cyclin A, but you don't need to know specifically which ones are required for each phase, but just know that there's different types of cyclins for each phase. Cyclins are regulated by their concentration. That's kind of the only real way they're regulated. So ubiquitin-mediated proteolysis leads to its destruction. Cyclins pull back the T-loop on CDK to expose the ATP binding sites, and that is what allows CDK to phosphorylate the proteins. So it kind of goes back to the idea of when something gets phosphorylated, it changes shape and it reveals something else for the next step to happen. Okay, and this is the graph that I was talking about with the different types of cyclins and their concentration during each phase. So CDK functions throughout the cell cycle with different cyclin partners, but for the most part, if you could see, like the G1 cyclin remains kind of constant towards the middle, but like the G1, the S and the M do change a bit more rapidly. And the next point is kind of the same thing with cyclin, provides specificity. That's how the same CDKs target different proteins in each each phase of the cell cycle. And then both CDK and cyclin contribute to binding interaction to the target substrate. So just kind of like I said before, you need cyclin and you need the CAC, how one is necessary but not sufficient. So the same way here, you can have the CDK activating kinase, but without the cyclin, it won't like direct it where it needs to go. So this is kind of a summary of everything we just talked about. So that CDK can regulate particular proteins in particular phases of the cell cycle, provide specificity. CDK must be phosphorylated by CDK activating kinase, and it's inactivated by the Wee1 kinase, which, like I said, we'll go over more in next week's topic because it's more relevant for the M phase. But and CDK inhibitors are typically seen in G1 regulation, a response to DNA damage. So that's going to go into P53. So P53 isn't the protein itself that's inhibiting the CDK. It's just a transcription factor, which binds to the DNA. So P21 can be translated and transcribed. So P53 is a transcription factor and tumor suppressor that responds to DNA damage. It activates P21, which inhibits CDK. If DNA damage is irreversible, P53 can also signal for apoptosis, and P53, once produced, is heavily regulated or degraded if inactive. So P53 is constantly degraded because if it was like always around, then the cell cycle would always be put on pause, so to speak. And that's all the content for this half. Is there any questions regarding to the content before problem sets?
Sure.
Questions?
Chat real quick. Okay. These for set.
Okay. Thank you guys for coming.
Thank you.
Just hit stop share and then